Yes, Dr. Gulani, please start.
Good morning to all. Welcome to everyone joining today's webinar. I am Dr. Pradeep Golani, and as the co-convener of this event, I am delighted to welcome such a diverse and engaged audience. Your participation greatly enriches our discussion and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey. SPSI engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I'm confident that today's session will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us today. We are gathered today to commemorate the 71st SPSR International Webinar on Natural-Based Polymeric Delivery System, Enhancing Therapeutic Efficacy in Pharmaceutical Science, a distinguished event in the series hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize the conferences that support the pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is the commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Dr. Najwa Mohamad, Associate Professor, Department of Pharmaceutical Technology and Industry, Faculty of Pharmacy, University of Cyberjaya, Selangor, Malaysia, as a distinguished speaker. Dr. Najwa Mohamad expertise greatly contributes to our event, providing substantial knowledge and insights. Her participation is certain to enrich our discussion and expand exploration of natural-based polymeric delivery systems. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the National Secretary of SPSR and the Managing Editor of both IJPSR and IJP. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. I am pleased to invite Mrs. Monika Sabharwal to warmly welcome our distinguished guest speaker, Dr. Najwa Mohamad. Over to you, ma'am.
Thank you, Dr. Pradeep Gulani. Hello, everyone. I am Monika Sabharwal, the convener of today's 71st SPSR International Webinar, and it is my pleasure to welcome you all to this enlightening session organized by Society of Pharmaceutical Sciences and Research SPSR. Our topic for today, natural-based polymeric delivery systems, enhancing the therapeutic efficacy in the pharmaceutical sciences. Explore the remarkable ways that natural polymers can improve the drug delivery, a subject of great significance in today's pharmaceutical landscape. We are honored to have an esteemed expert with us, Dr. Najwa Mohamad, Associate Professor in the Department of Pharmaceutical Technology and Industry at Faculty of Pharmacy, University of Cyberjaya, Malaysia. Dr. Mohamad brings a wealth of experience in the innovative use of natural polymers for drug delivery, and her insights will surely provide our attendees with a valuable knowledge on improving the therapeutic efficacy through these advanced systems. I am pleased to share that today's event has drawn more than 1,800 registrations from over 27 countries, demonstrating the global interest in this field. As we celebrate the 71st SPSR International Webinar, we are incredibly grateful for the support we have received from the participants worldwide. Your positive feedback motivates our team to continue providing the high quality content and insightful discussions in the pharmacy profession. Our SPSR webinar series has been steadily growing, bringing a diverse and relevant topics that are often chosen based on our audience suggestions. This approach helps us to meet the educational needs of our community and ensures that our webinars remain dynamic and very impactful. We are also excited to announce the SPSR Webinar Membership Program, allowing the participants to register just once for the entire year and gain uninterrupted access to all the webinars. The need of the multiple registrations is not needed. In addition, we invite the national and international institutions to collaborate with us for the future activities. Any interested institutions are welcome to reach out with the proposal for the collaboration, as SPSR is committed to foster a collaborative environment that advances the research, education, and innovation. At SPSR, we also continue to work hard in bringing together the prominent speakers from around the globe, facilitating a knowledge sharing on the various areas of the pharmaceutical sciences and healthcare. This series would not be possible without the tireless efforts of the SPSR team, whose dedications allow us to deliver enriching content every Sunday, creating a positive impact on our audience and reinforcing our mission to elevate the pharmacy profession overall. Before we commence, I would like to warmly welcome Dr. Shashi Alok, our National President, whose visionary leadership has been very crucial in impressive transformation of SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Dr. Pradeep Golani, the co-convener of this webinar, and the organizing committee members, Dr. Padmani Shukla, Mrs. Swati Jain, Dr. Saurabh Kose, Dr. Sanjay Nagde, Dr. Pradeep Golani, Mr. Ankur Agarwal, Mr. Junaid Tantre, and Mrs. Nidhi Mittal. Their tireless efforts have played a crucial role in transforming the vision of this event into a reality. We thank to each one of you for joining us today and for your continuous support, which has helped the SPSR community to grow and thrive. We are confident that today's session will deepen your understanding of natural-based polymeric delivery systems and their potential in enhancing the therapeutic efficacy. Keep up with the innovation through the SPSR. Subscribe to our YouTube channel for exclusive webinars, expert insights, and the latest research in pharmaceutical sciences and healthcare. Join our dynamic community contributing to healthcare advancements. Stay updated by subscribing to SPSR YouTube channel and enabling the notification bell icon for the instant alerts. Follow our journey in advancing the pharmaceutical sciences for the societal benefit. Once again, thank you everyone for your continuous support, and the special thanks to those who regularly attend us webinars on Sunday. Your participation keep us motivated and help us to improve the every session we organize. Without further ado, I am pleased to welcome Dr. Najwa Mohamad ma'am, who will lead us through this very fascinating topic. Over to you, Dr. Pradeep Galani.
I appreciate Mrs. Monika Sabharwal for granting me the opportunity to present this webinar. And now we will start with a short introduction to guest profile. Dr. Najwa Mohamad is a leading figure in pharmaceutics and pharmaceutical technology with a robust career combining academic excellence and industry expertise. Currently an associate professor at the Faculty of Pharmacy, University of Cyberjaya, Dr. Najwa holds a PhD in pharmaceutics from National University of Malaysia and has become widely recognized for her research contributions in drug and cell delivery systems, wound care innovations, the halal pharmaceutical industry, and the development of biodegradable films. Her work has been instrumental in advancing sustainable and ethical pharmaceutical solutions, and she has contributed valuable insights into the integration of halal compliance within the pharmaceutical technology. Dr. Najwa's innovation research has garnered numerous prestigious accolades, including the gold medal at the Innovex 2024 and the silver medal at both the International Invention, Innovation and Technology exhibition and the International Medical Education Conference in 2023. These awards reflect her commitment to pushing the boundaries of pharmaceutical technology and her skill in translating complex scientific concepts into practical, impactful applications. Beyond her research, Dr. Najwa has a rich background in industry roles. As a former product development manager at the Pharmaceutical Innovation Commercialization and Service Center, she played a key role in bridging the gap between the academic research and commercial product development, overseeing projects from concept to market ready solutions. This role provided her with the unique insights into the demand of pharmaceutical commercialization and equipped her with a strategic approach to product development. A dedicated educator and mentor, Dr. Najwa is deeply involved in guiding the next generation of pharmaceutical scientists, offering her expertise through lectures, hands on training, and individual mentorship. She has published extensively in reputable journals, contributing to the global dialogue on pharmaceutical advancements and remain a committed advocate for the continuous evolution of pharmaceutics. Her work is both locally and internationally recognized, making her as a prominent voice in the field and a vital contributor to the future of pharmaceutical sciences. It is with great satisfaction and pride that we declare that we have exceeded 1,800 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from nations including India, Afghanistan, Algeria, Azerbaijan, Bangladesh, China, Egypt, Germany, Indonesia, Iran, Iraq, Malaysia, Myanmar, Nepal, Nigeria, North Macedonia, Oman, Pakistan, Saudi Arabia, Sri Lanka, Syria, and UAE. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an enlightening and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Najwa Mohamad, with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Thank you. Right, is it my slide is already there?
Right, ma'am. You are
share,
audible and right.
Okay. Okay, first of all, good morning to all the participants for this webinar, participant of the Society of Pharmaceutical Science and Research webinar. And I would like to say thank you again to our moderator, Dr. Pradeep Golani, for the introductions of myself and Dr. Monika for the introduction of this webinar. Okay, brief introduction will be given by Dr. Pradeep Sukolani, and my research interest is regarding a natural-based polymeric drug delivery system, a cell delivery system, and I also is an expert for the hydrogels, development of the hydrogels, and they have the various of the polymeric delivery system that we made into the dissolvable films, hydrogels, and now currently my focus research is also on the halal industry. So we go back to the webinar topics is on the natural-based polymeric drug delivery system, enhancing therapeutic efficacy in pharmaceutical science. And I'm in the Department of Pharmaceutical Technology Industry in the Faculty of Pharmacy, University of Cyberjaya. And a bit brief of the University of Cyberjaya is very near to, it's in Selangor, very near to the Kuala Lumpur, the capital city of Malaysia. And KLIA is actually very near to the University of Cyberjaya. Okay, without further any delay, I will start my webinar with the introductions of the topics. Okay, all right. So for first of all, the definitions of the polymer. Okay, when we say about the polymer, we know that is a big molecules. Is a macromolecules made of a simple structure unit. So they are monomer that are actually linked together, whether it's through the covalent bond, or we can say that from the hydrogen bond is made up of a simple structure unit of the monomer, and it become a polymer. And then this polymer, we are mentioned that as a macromolecules. And this polymer, we can actually categorize it as a natural or synthetic base of origin, whether it's a natural base or synthetic base of origins. And there are different source of nature. They can be from the plant source, it can be from animal source, or it can be from the microorganism source. And synthetic base of polymer, it can be from the reaction of the chemical itself. In terms of the characteristics of the polymer will be different. Structure, in terms of the structural, the mechanical and functional properties can be modified, and blending with the various functional groups. Let's say we want to modify the structure of the polymer or the mechanical strength. Let's say we want to increase the strength. We can modify with the functional group of the monomers, or we can actually graft it with the monomers that actually have a different function of the chemicals. Let's say they have the OH group or carboxylic groups. We can actually modify the structure and mechanical. That's why a polymer is widely used, because it are very flexible. In terms of widely used, is very flexible, that we can fine tune, or in terms of we can actually modify the structure and mechanical strengths. And in terms of the application of the pharmaceutical science, what is the application that can be used for the polymers? There are various, there's a lot, and we know that it's high being used in our daily life. So there's a lot of polymer that been used, like our daily life, that we go to the mall, we use a plastic, so that is a polymers. And also in our body also, they have an embedded of the polymer in our body. They are DNA, they are proteins, all are called they are called polymer as well. Okay, and the application that we can use is in terms of pharmaceutical science. Of course, we want to use it as a drug delivery system for site-specific targetings, and it acts as a control membrane to release the drugs. And also we can actually sustain the drug release, so we can modify it on the intended use, so we can actually control it, or we can target in the specific area or a specific organ based on target organ. Okay, whether we want to have a very we can say a very short degradables of the polymer, or we can say that it's non-degradable polymer that actually can embed it in the cardiovascular, or we want to implant in our tissue system. We don't want it to be degradables. So they have a different type of polymer that can actually be selects for the intended use. Okay. And then other than that, we can actually stabilize, we can protect the drugs We can encapsulate the drugs, and also we can mass taste the drugs. Let's say the antidepressant drugs, some of the drugs that are very bitter in the taste, so we can mask the drugs with a polymer for the oral drug delivery system. Other than that, we also can modify flow characteristic in the liquid, or whether in the liquid or solid dosage form. It either it depends on the targeted area and what is the drugs and what is the characteristic of the drug itself. Okay, before we go further, we want to know what is the history, how actually the polymer is being developed. It's been developed since beginning of our life, like I mentioned. The biopolymer is already in our human's body, like DNA, RNA. Like DNA is a cross-link with the covalent bond, so it's already there in our daily life, in our body. And polysaccharide has a crucial for metabolic function as well. You know that. And natural polymer have been used for centuries to make goods such as clothing, utensils, and also decorative items. In the 18th century, they started to improve the properties of elastic material by adding specific additive. And then in 19th centuries, they start to use a tire. They start to implement, okay, what is the tire that we use until now is a Goodyear. Until now, we will still use the Goodyear. Okay, it enhances the elasticity of the materials, and then they have a revolution on the polymer industry, begin to modification or in the chemical structure and the polymers. And the early 20th century, they are the first synthetic polymer that been developed. So we know that back then is a natural polymers, and recently there are synthetic polymers so that been developed. And also Hermann Staudinger in 1919s, they are systemic studies of polymer science. They identified that the polymer consists of long chain, and how actually they are consists of the long chain, how they actually link one to another is by the covalent bond hydrogel, covalent bonded molecules. So it's either covalent bond or hydrogen bonds is there actually. So they already find out that, oh, there's actually there are a polymer that actually been linked together by the monomers. Okay, next, the advantages of a drug delivery system is, if you want to say about the advantages, of course, we want to say advantages of natural based polymer, regardless of the we compare with the synthetic polymer. So what is the advantages of natural based polymer? Of course, the first and foremost is biodegradable because it's easily degraded by and metabolized by the enzyme. Of course, when they have the hydroxy groups in the polymers, so they can actually absorb the water and they can actually swell, and then after that they can degrade and leave no residue and making it environmental friendly. But there are also natural based polymer that are not degradables. So we are not mentioned that all the natural based polymer are degradables, but certain polymer, even though they are natural based, but when like humans, right, cellulose, when they are ingested by humans, some of because we don't have the enzyme cellulase to actually digest the cellulose, that's why some of the natural based polymer, they actually cannot degrade. And in terms of biocompatibility, yes, they have a lot of the advantages compared to the synthetic polymer, because we know that biocompatible in terms of their chemical structure and structure of the natural base itself, it make them is very compatible to human body. And we know that the biocompatibility effect is because the polymer derived from the common monomer in living organism and minimize their toxicity. And in terms of economics, of course, we can say that in terms of economic, they are very cost effective because it's easily available. Like in my research, we use like a food grade product, like we use a dessert like Natalie Coco, and we use a physium hearse. So it's easily and readily available as a food grade to make into the polymers, to make into the membranes. And other than that, they use a waste. They can use a banana peel waste. They can use a fruit waste, like the skin of the fruits. So they can use all these waste like palm waste. So all these waste actually can actually extract the polymer from the waste materials and form the we can say that form polymeric membranes for drug delivery or others applications. In terms of versatility, yes, in terms of versatility, this is a crucial part of why polymer is widely being used because it's versatile. Anyone can actually adjust it according to what they intend to make the polymer for. So if they say that they want to actually increase the degradable time, so they can actually tailor the drug delivery solution by actually choose the polymer that actually slowly degraded. So it can actually versatile the structure and molecular structure and also the functionals and also the properties of the polymer itself. Okay, when we talk about the source, there's a lot of source of polymers. They can actually have from the plant-derived materials. This is when we say that it's the natural polymers. They can have the cellulose, they can have the psyllium husk, they can have the starch from the plant-based materials. Other than that, there are animal-derived materials such as the collagens. Okay, the collagens from the animals. Okay, they can actually extract the collagens from the crabs, chitosan, and also the wools. And the mineral derived material, they have a silica, natural salt, and also in addition, they also have the microbial origins. They also have the microbial origins that are, we say that polysaccharide like alginates, bacteriocellulose is one of the example of the natural polymer of microbial origins. Okay, when we say about tailoring the polymer properties for targeted delivery, yes, it's for the controlled drug release because we can select the modification, selective modification of the polymer structure and functional group enable the target release. Whether we want to actually graft it with the polymer that are containing a certain structure like carboxylic acid with the monomer that have the hydroxyl group, so we can actually tailor and modify the polymer structure. In terms of the formulation optimization, we can actually adjust the parameter. Like we want to adjust whether we want to develop the nanoparticles or whether we want to develop the membranes, polymeric membranes for a nano size, a micro size, or a macro size. We can actually adjust the formulation, and we can adjust the charge and also the hydrophobicity that can enhance the lipophilicity that enhance the tissue specific uptakes of their polymeric membranes. And also we can adjust on the stimuli responsive behavior, whether we want them the polymer to swell in the acidic structure, acidic fluids, acidic environment in the stomachs, in the gastric fluids, or we want it to swell more in the intestinal fluid. So we can actually tailor and engineer and modify the structure to respond to physiological triggers, such as we want to avoid the degradations of the polymer from too low of acidic like pH 2 or pH 5, or we want it to be digested by the enzyme, or we want to protect it to be digested from certain enzyme. Okay, then what is the innovative polymeric materials that actually already been produced and already conducted a research? Nowadays, there's a lot of innovative polymeric materials. Okay, one of it is nanosystems, and also they can use for coating the drugs, and they can actually form films or membrane that have composite films and membrane. They can embed it, the drugs, they can embed the DNA, they can embed it, like let's say they can embed the nucleic acid, they can embed the cells inside the composites. and delivery. And also microneedles. When we say about the microneedles, usually the recently of the microneedle, the characteristic, they want it to have the sustainable research, that actually they prefer the biodegradable microneedles, that actually when they are embedded in the skin or through the transdermal, that actually the polymer can actually dissolve and while deliver the drugs through the transdermal drug delivery. Other than that, other than pharmaceutical sciences, they also have their water remediations. And in terms of biologicals, biomedical terms, they are also the wound healing and drug delivery system. So for the wound healings, they can be used for the topicals to deliver the drugs in the ulceration in the wound healing process. And also there are packaging, active food packaging. We know that polymer is widely being used to use for the packaging materials. So they use a natural based polymer because they want it to be degraded. So there's a lot of we can say that innovative polymeric materials for advanced application have been used nowadays. And what I want to talk about in detail today is an application of natural-based polymer that my research group have been done. That there are two types of the polymer that I will focus today. One type of it is a bacteriocellulose-based polymer. This polymer is actually origin from the microbial microorganism, which is a bacteria. And also the next one is a plant-based source. Okay, there are arabinoxylum-based polymer. They are actually from the plant-based, we call it as a Physaelium hars. So when we talk about bacteriocellulose, in a research study that we done in our laboratory, so we actually extracted the bacteriocellulose from a dessert that we call it a nata de coco dessert. Is a jelly-like structure. Okay, nata de coco is like a jelly-like structure that we extract the bacteriocellulose from this jelly, and we use it. We extract the polymer and we grafted it and form a hydrogels to be used as topical treatment and also cell delivery. And other than that, we also have the arabinoxylum that we extracted from the Physaelium hars. Physaelium hars is, we know that is a cereals, okay, is a polysaccharide. It's being used widely for a digestions, a good digestions, for a good, we can say good for gastrointestinal tracts, because it can relieve the constipation and also diarrhea. And we use that as actually for a drug delivery. And currently we study on the dissolvable film of the arabinoxylum from the Physaelium hars. So we will go details about the study. Okay, the first and foremost, like I mentioned just now, we use the polymer. There are natural source from microorganism. They are bacteriocellulose. They are produced from the Acetobacter xylinums. So how this jelly is produced? Actually, this jelly is originated from the Philippines. So this jelly is produced by the fermentations of the bacteria that ferments the water, coconut water. So they fermented the coconut water, and they form a cellulose from the fermentation process. So they ferment the glucose and they produce a cellulose. So we get a cellulose from the jelly, this nata de coco, right? And then we use it to form a hydrophilic 3D structure that can absorb large amount of water. And then we use it as a hydrogel wound dressing, and then we fine tune to actually increase the mechanical strength. And they are also have the very huge capability or capacity to absorb large amount of water. And how we produce the hydrogen membrane, the polymeric membranes, is by grafted with the acrylic acid. We use a monomer that is widely used for synthesis of the hydrogels. So the acrylic acid is being used to grafted with the bacteriocellulose to form the hydrogels. So first and foremost, when we have the nata de coco from the supplier, we purify it first. We make it into the pH of 7.4, and then we make into the smaller particles. We grind it into the smaller particles, and we freeze dry into the powder, and we are mix up with the acrylic acid and form the solutions, and we actually use the electron beam irradiations. So we have the dispersion of the combination of the bacteriocellulose and also the acrylic acid, like this one, and we grafted by using the electron beam irradiations. In Malaysia, we have the Malaysian Nuclear Agency that actually have the facilities for electron beam irradiations. So without using the cross-linking agent, we use this to irradiate the hydrogels and form the hydrogels. And then after that, we further study the effectiveness of these hydrogels. And I mentioned we can actually modify and we can actually formulate the hydrogel in terms of the concentration of the ratio of the acrylic acid and bacteriocellulose. So we adjust, we modify on the concentration of the acrylic acid and bacteriocellulose, and then we irradiate it by the electron beam irradiation, because we want to graft the monomer. We want to graft the acrylic acid monomer with the polymer of the bacteriocellulose. So usually the bacteriocellulose, they have the hydroxy groups in the structure. And then in terms of acrylic acid, they have the carboxylic group that actually can enhance the grafting of the bacteriocellulose and also the acrylic acids to form the hydrogels. Okay, so we confirm that by using the FTIR, when we finalize on the hydrogel developments, we go for the FTIR. So we check that I mentioned that the combinations of the groups of in the BC that are hydroxy group and the carboxylic group of the acrylic acid, we form the hydrogels. And after that, we go for the compositions and also the characterizations of the different formulation of the hydrogels. And we can see that in terms of when we increase the concentration of acrylic acid and when we reduce the bacteriocellulose concentration, the BC concentration, we found out that the thickness is increased together when we increase the dose radiations, and also the water vapor transmission rate also are decreased, because we know that when we increase the dose of radiations, when we increase the acrylic acid monomers, and then we know that there are cross-linking increase in the cross-linking density, we know that there are increase of the polymerization occur. So that's why they are a bit tight and then they are dense. That's why we can say that the pore structure are very less, then it causes the water vapor transmission rate a bit lesser compared to the others. There are also a significant difference compared to the other compositions. In terms of the mechanical strength and elongation, also we see that the higher the mechanical strength is, we can have a look at the concentration of 40% of the acrylic acid and 60% of the bacteriocellulose. And we can see that it increased their tensile strength, it increased their elongations. And in terms of the swelling ratio, we see that when there are lower concentration of acrylic acid that grafted to the polymers of the bacteriocellulose, there are a lot of porous structure. There are a lot of porous structure, and the porous structure are bigger. That's why they can absorb large amount of water, and they are we can say that the swelling ratio is higher. They are swelled more compared to the higher concentrations of the dose of the bacteriocellulose and higher dose radiations when we say at the 50 dose radiation. So we can say that the swelling ratio a bit lower compared to the lower concentration of the acrylic acids. Okay. And then next, we see on the composition and characterization of BCAA. We see that the hydrogels, the percentage of swelling is lower, okay, when we increase the concentration, and also the equilibrium water content also is lower when they are increased in their irradiation dose. What we can see under the microscope of the hydrogel, we can see that they are different in the structure. The porous structure is smaller, and the concentration of the acrylic acid is more compared to the lesser concentration of acrylic acids. And when we say that higher polymers and lower concentration of acrylic acid, and also we can see that there are significant difference of the porous structure. They are more in the lower concentration of acrylic acid compared to higher concentration of acrylic acid. In terms of when we see the difference, we want to see whether they have the effect on the reiteration dose or not. While we maintain the concentration similar, but we want to see whether dose radiation play a big role or not on the swelling and water retention. And the result demonstrate that the swelling ratio is higher in the lower dose radiation. So means that when the monomer percentage is lower, and then together with the concentration the dose radiation also a lower, we can say that the swelling ratio is larger. And then how do hydrogel interact with the cells? We want to know whether the hydrogel is compatible. As mentioned, that natural base polymer is very compatible because they are from a natural source, but still we need to actually test whether it's compatible to the red blood cells or compatible to the fibroblast cells. So we done on the cell toxicity, and we use a live death fluorescent image to see the effects of the cell that been treated with the hydrogels. And we see the result that are 80% above of the cell ability, and is being confirmed by the microscopic observation of the live and death fluorescent image of all the concentrations of the hydrogels. And in terms of the hemolysis of the hydrogel, we can say that there are no hemolysis occur, and the hemolytic percentage or index is very low in the hemolysis of hydrogel study. And this is a image that we see that the cells attached on the top of the surface of the hydrogels. We see that the round shape there is the cells. And we also do the survivability test on the different dose radiation. We want to see whether there are effect on the dose radiation when we have the constant concentration of the BC and AA. So we have the indirect method to see the solubility, and also we have the direct method. Direct methods mean that we directly study the hydrogel on top of this on top with the cells, and indirectly usually we embedded the hydrogel in the media, and then we take the media and test on the cells toxicity studies. And the highlight of that finding, we want to say that when we increase the concentration of acrylic acid and irradiation dose, is increase the thickness and increase the cross-linking density, and is improve the mechanical properties of the hydrogels. And the spreading capacities of the hydrogel increase significantly with decrease in the acrylic acid composition in simulated wound flakes. And we see that they are not toxic to the fibroblast skin cells, with the webbability is more than 80%, and is found to be biocompatible with the blood with low hemolytic index from 0.8 to 1.3%. After we find out the in vitro studies on the characteristic of the hydrogel, we intended to study the effectiveness of these hydrogels to be used in wound healings. So we know that the process of the wound healing, there are 4 stages, and all the 4 stages is very crucial to determine the wound healing process. And the first and foremost is hemostasis, whereby we want to have the blood vessel is constricted, so we know that all the platelet will be embedded in that area of the wounds, and then they are together continue with the inflammations process where we want neutrophil to be attracted to the wound healing area. And then next is a proliferation process, where we want to have the formation granulation tissue by the epithelium to cover the wound beds to decrease the scalp area. And also in the remodeling process, we want to actually increase the collagen level during the remodeling process and increase the strength of the wound itself. So we did a study to identify whether they are have a potential or not in the wound healing. Yes, we have the positive results to actually find out that the hydrogels are they are significantly different compared to untreated wounds. So we know that with the hydrogel itself, they are actually have the effects to provide the moist environment to the wound healing and enhance the wound healing process. And we also observe on the microscopic observation by study the mesenteric and also H&E. For this one, we see that in the day 14th, for the treatment of the hydrogels, there's a lot of concentration of the collagens, and we see that there are epidermals, we can say the thickening of epidermis layer. And the next, not only we stop there, we also go for the cell delivery effects of the hydrogel. We want to know whether the hydrogel can actually deliver the cells. So the hydrogels we use is 3D hydrophilics, that cross-link polymeric networks. We know that actually they are 3D scaffold, and it can actually embed the cells. The cell can embed it into the polymeric membranes. And the cell that we use is keratinocyte cells and fibroblast cells. And we want to use whether it's possible or not to induce to actually accelerate the wound healing process, and also they are either they are biocompatibles and not toxic, and also we know that in vitro there are porous structure inside the hydrogel, so we want to know whether it's possible to embed the cells into the scaffold and deliver it to the wound bed. And then how we studies on the hydrogel itself to deliver the cells. Of course, we want to know whether the cell is very compatible for the hydrogel itself. So first and foremost, we see on the cell attachments. We want to see whether the keratinocyte cells and fibroblast cell can attach or not on the hydrogels. And then after that, we want to know whether the cell can migrate. Is there any movement of the cells into the porous structure of the hydrogel, because we want it to be transferred from the scaffold to the area of the wound bed. So we how we do the cell migration study is we embedded the keratinocyte cells and also the fibroblast cells, and we dye with the different color, keratinocyte cell with the green color and the fibroblast cell with the blue color. And then after that, we also want to study whether the hydrogels can enhance the transfer of the cells to the other layer. Okay, because we want the origin, the idea is we want it to be transferred from the scaffold to the wound bed area. So how we do is we use the staining as well. We staining the keratinocyte cells with the green color and also the fibroblasts with the blue color. And then we have 40 case cells here for the keratinocyte cells, and for fibroblast cell we have 40 case cells, and we embed it into the hydrogels. So when we embed it in the hydrogel, we wait for a few hours, and then we see on the 3 days view, we see that whether it's already transferred to the ovine collagen hydrogels or another type of hydrogels. So we use the ovine collagen hydrogels, and then we want to see whether the cells is being transferred from the BCAA hydrogel to the ovine collagen hydrogels. And also we want to see whether this hydrogel is very compatible to the different type of cells by using the alarma blue and also live dead cells. And how we actually get how actually the source of the keratinocyte cells and fibroblasts is obtained. So we conducted this in the hospital under National University of Malaysia back then. So we harvesters and construct the cell carrier. So first and foremost, we use the patient's skin. So the patient's skin from the hospital, we take it and also we digest it with the collagenist, and then we collect the keratinocyte cells and fibroblast cells from the patients. And how to differentiate and to actually separate the keratinocyte cells and fibroblast cells, we use the trypsinizations. So from this trypsinization, we can separate the fibroblast cells and keratinocyte cells. So we grow the fibroblast cells and also we grow the keratinocyte cells based on the amount that we want to actually embed it in the hydrogels. So after we have fully grow the keratinocyte cells and fibroblast cells, we actually construct the cell carrier. So we embedded both of the cells on the cell carrier, which is the hydrogel from the petrocellulose-based polymer. And what if we find out that the cell attachment is increasing? Cell attachment is increasing up to, we can say that after at 4 hours, there are more than 80% of fibroblasts and keratinocyte have attached. And this is a morphology structure that we see that they are embedded of the fibroblasts on top of the hydrogels. With regard with the cell transfer, there are two type of the hydrogel that I mentioned just now. We have the petrocellulose hydrogel and also we have the collagen hydrogels. We want to see whether there are cell transfer occurs in between the petrocellulose to the collagen. Yes, and then they found out that they are cells actually embedded also in the collagen in the group of fibroblasts and keratinocyte cells. So we confirm that they are a transfer of the cell from the BC to the collagen, and we know that they are a migration of the cells towards another type of hydrogels. So we want to confirm. We do the fluorescence. Okay, in the fluorescent we use the fluorescent microscope, and then we quantify the numbers of the cells. We can see that the percentage of the hydrogels in the beginnings will be higher, and then after the day 3, we can say that the beginning will be higher in the collagen level, right? And we can see that from here there are a migration. You can see that the moving of the fluorescence in the green color there. So we know that there are a migration of the cells into the porous structure of the hydrogels. We also try to study whether there are effects of the hydrogels with the cells comparing with the untreated and only the hydrogel alone. We know that when we embedded with the cells, there are a better effects compared to the hydrogel alone and also the control, because it can increase the wound closure measurements in the after the day after 2 weeks times after we give the treatments. So basically how we do that is actually we embedded the hydrogels on top of the nude mice, and then we suture the construct criteria of the hydrogel with the cells, and then we suture on top of the rats. And then after 13 days, we see that the hydrogels is actually induce the wound closure, and the size of wound closure is reduced, and we can say that the scalp also reduction of the scalp is faster compared to the other groups. And this is histological observations from the H&E and also the mesenteric. We can see that the H&E for the hydrogel with the cells, we can say that they are intact of the epidermis and dermis, and we can say that collagen mesotrichome staining of the collagen is more. Yeah, we can see that in group of the hydrogel and cells. Okay, Okay, next is we want to see on the normal skins effects with the hydrogels with the cells. We want to see whether there are various observations. Okay, we can see that there are a marker, involucral marker for the epidermis. We want to see whether the dermis layer is actually enhance the proliferation and also collagen level and also alpha small muscle to produce more fibroblasts. there are positive results when we say that the hydrogel with the cells, because we can see that there are increased amount of the collagen type 1 and also alpha small muscles. And then next is arabinoxylans based hydrogels for drug delivery. This is another we can say a polymer other than the bacteriocellulose. We also study on the arabinoxylan that are produced from the Physacium hars. So the arabinoxylans are taken, extracted by from the Physacium hars. And it show the physical chemical properties that are very soluble and also can enhance the viscosity and is able to form the hydrogels. And together we also grafted with the acrylic acids. Is a monomer of the acrylic acid. So we grafted the arabinoxylan with the acrylic acid, and we want to form the hydrogel by using the electron beam irradiation, because we want to reduce the harmful risk without using any cross-link agent. Can I take some time to actually get the battery into my laptop?
Yes, you can take, ma'am. You can take.
So sorry for the trouble. Can I continue now?
Yes, ma'am. Please go ahead.
Right.
Okay. So I mentioned about the bacteriocellulose just now. Other than the bacteriocellulose, we also use the arabinoxylans polymer to form the hydrogels. So in this study, we use arabinoxylan and also the acrylic acid. Okay, we use the arabinoxylan and acrylic acid as a monomer, and acrylic acid is widely known to actually with the carboxylic groups to be used to form the hydrogels. And with the acrylic acid, so it can actually enhance the stimulant responsive of the hydrogels. So the hydrogel is consists of a hydrophilic component that can actually absorb large amount of water, and also they have a mimics extracellular matrix, and it can be classified into reversibles and permanent hydrogels. And in terms of the development of the hydrogel, we use electron beam irradiations. And how actually my students actually extracts the arabinoxylans. Okay, they use a water extraction method. So actually they extract the arabinoxylans, and by using the physalim hearts. The physalim hearts actually they get from the food source, food grade, and then they extract the arabinoxylans and by using the water extraction methods, and they collect the arabinoxylans and then they synthesis it and make it into dispersion of the arabinoxylan and acrylic acid to form the hydrogel with similar ratio, but in terms of different dose radiations. And after that, they do the characterizations of the hydrogels and they study on the physicochemical properties such as the FTIR analysis, the SEM analysis to see the morphology of the hydrogels and also the swelling test by different pH in terms of different pH and also different temperature, because they want to actually see the responsive of the hydrogel in between of a different pH and also different temperature. And also they also loaded the polymer with the model drugs. They are paracetamol that being used as a model drugs, and then they see the in vitro studies released in the different pH at a temperature of 37 Celsius, and they actually okay. Okay, pointer. Okay, they actually studies the in vitro release by pH of 1.2 and pH 7.4 in the temperature of 37 Celsius. And then they also study the cytotoxicity on the fibroblast cells. And the results show that the percentage of yieldings of the arabinoxylans from the Physalim hearts is more than 50%. So we can say that Physalim hearts can be considered as a good source of arabinoxylan. And then for the hydrogel itself, this is how it look like. The hydrogel is formed, and the hydrogel is successfully synthesized via the electron beam irradiations. And we can see that the 30-70 concentration of the acrylic acid with the arabinoxylan with dose radiation 50 exhibit the most solid and durable network due to the higher radiation dose. And the physicochemical properties of the hydrogel can be modified by varying the electron beam irradiation, whether we want to make into the lower dose radiation or we want to increase the dose radiations. And in terms of the gel fraction is to determine the degree of cross-linking within the hydrogen networks. So you observe that when we increase the dose, radiation dose, the gel fraction is increased. So when the gel fraction is increased, we know that they are signifying the higher degree of cross-linking between the networks. And hence, we know that hence gel fraction has a bit an inverse relationship with the swellings. And this is a visual examination. We can see that when we dry the hydrogels in the drying ovens, we can see that it's a bit yellowish, but when we swell in the distilled water, they are become more colorless. And the thickness of the hydrogel, when we see that the higher dose radiations, okay, they are causing the hydrogel more thicker, okay, because we saw that is indicating that it's dependent on the radiation dose. So the hydrogen thickness indicates that it's very dependent to irradiation dose. A higher irradiation dose result in higher degree of cross-linking, so contributing to a thicker, more rigid network, and they see that the pore structure is very smaller. And then we see that in terms of the pH determinations, we can see that the results show that the pH of all the hydrogel varies from the around 5, pH of 5, which significant decrease with increasing radiation dose. We know that the slight acidic properties of the hydrogel are due to acrylic acid. So when they are less grafted of the acrylic acid on the BC on the arabinoxylan, we see that there's a lot of presence of carboxylic acid functional group. Hence, it's actually increase the the pH. And we can confirm that is have the grafted of the hydrogel when we do the FTIR of the arabinoxylan and also acrylic acids. right? And the FTIR analysis show that the bending, show the C double bond O bending and C double O stretching at different timelines, okay, and then we can say that they are present of the C bond present of any new peaks at the 430, 70, 35 hydrogen spectra indicate successful cross-linking during hydrogen formations. We see that mostly I mentioned about the COOH of the acrylic acids and C double bond O of the hydrogels is important to actually grafted the BC and also the arabinoxylans. And in terms of the structure analysis of the SEM structure analysis or macroscopic observation, we can see that when we increase the dose radiations, the pore structure become very small compared to the lower concentration of those radiations. So the F30, 70, 35 show the biggest pore size with a range of 116.7 to 647.9 micrometer. And the pH, okay, we can see that the swelling, okay, the swelling of the hydrogel in different pH. In the pH of 1.2 and also pH 7.4. We want to see which hydrogel with the different dose radiations swell more. So we can see that in both acidics, or we can say that neutral pH, the hydrogel with the lower radiation dose is swelled more. Okay, the swelled more because we know that the degree of cross-linking density is actually have the effect on the C double bond O group to ionize. And in terms of temperature, we see that in the room temperature, there's not so much difference between different type of hydrogel. But when we see on the room, in the human body temperature, okay, we can see that there are more swelling. Okay, so we can say that in the lower concentration of dose radiation, it has a bit the higher degree of swelling as it can swell more readily during the water absorption phase as compared to the other formulation. And for the drug loading, we know that the 30, 70, 35 is the most well. So we choose that hydrogel to actually use to load it with the paracetamols, and we see that the percentage of loading of this hydrogel, the drug to the hydrogel is 96.83%. And in vitro release show here, there are controlled release at a pH of 7.4. And we can see that based on kinetic model, the calculations, they are actually highest regression coefficient at the Coase-Meyer papers, that actually this hydrogel is fit the model of Coase-Meyer papers. And the cytotoxicity, we can say that are more than 50% of cells viable when we treat with the arabinoxylans hydrogels. And that is all on the arabinoxylan hydrogel. So now we have the industrial grants from the industry. We develops antimicrobial properties of arabinosodium arginate together with the Ganoderma. So we load it with the mushrooms. We want to see whether it's capable or not to actually have the antimicrobial effects. And also in the future pipeline, we also want to actually explore more on the composite dissolvable films for oral drug delivery or transdermal drug delivery or topical drug delivery. And this is a extraction of the arabinoxylan to actually produce the dissolvable films. This is extraction of difficilium hars, and this is a formulation that we use to produce the dissolvable films. And then this is a Ganoderma that we embedded into the to the gels, to the films. And then this is the film after being loaded with the Ganoderma. It become very brownish in color due to the Ganoderma color itself. So that is a overall pipeline studies, and that in the conclusion, I will say that nature-based polymeric delivery system, such as either bacteriocellulose, chitosans, alginates, or arbenoxylane-based hydrogels or polymeric membranes, demonstrate significant potential for enhancing therapeutic efficacy in pharmaceutical science, as mentioned in the in my my slide just now. And these materials are biodegradables, biocompatible, and can be tailored for targeted and controlled drug delivery applications. And recent advancement in hydrogen synthesis and characterization can highlight their versatility. So we can actually see on the structure modification on the functional groups, and we know that they are also a promising result in the cell attachment, drug loading, and sustained release. So continued research on the natural-based polymer offer eco-friendly, because mostly we know that the research on the natural polymer is actually being pushed, and then nowadays there's a lot of research of the natural-based polymer being studied, because we want to have the eco-friendly and sustainable an effective solution for future pharmaceutical innovations. And this is a publication that we sent to the Biomaterials Society for the Biomaterials. We published in the carbohydrates polymers, in the scientific report for the cell carrier cell delivery, and also we sent to the drug delivery and translational research. And we also a group of our studies also won an InnoVax in 2024 for the project of Indici Hills. We have secured the gold award, and this is my students that actually won the first place in the my PSA. My PSA is a society, student society of Malaysia's Malaysia Pharmaceutical Society. So she won the first place for the topic on the arabinoxylane hydrogels. I think that's all. Thank you for my talks. That is all for my webinar today. I'm so sorry for the technical issue just now on my battery of the laptop. I'm so sorry. Thank you.
Thank you so much, Dr. Najwa, for delivering an excellent session on natural-based polymeric delivery system, enhancing therapeutic efficacy in pharmaceutical sciences. You started this session introducing us about the polymer and its application in drug delivery system. After that, you discussed the chronological development of biopolymers and advantages of natural polymers-based drug delivery systems. You explained that natural polymers can be derived from the plants, animals, and it can also be derived from minerals and microorganisms. You also told that the targeted drug delivery can be achieved by tailoring the polymers. You introduced us with the various innovative polymeric materials like gelatin, alginate, chitosan, cellulose, etc. After that, you focused your talk on hydrogels formed from the natural-based polymers and how it interacts with the cells. You also talked about bacterial cellulose hydrogels as a scaffold for cell delivery. You explained us the process of wound healing and how effective are hydrogels in healing the wounds. You also discussed on different methodologies for preparation and characterization of hydrogels. Thank you, ma'am, for educating all our participants on this novel topic of natural-based polymeric delivery system. And now there are some questions from the participants, and I will be asking you one by one, ma'am. The first one is, ma'am, what are the challenges in scaling up the production of natural-based polymeric drug delivery system for commercial use?
Okay. First of all, I would like to thank you for the questions. In terms of the scale up, of course, in the development, because we use I mentioned we use electron beam irradiations. We use the electron beam irradiation. So the facilities is the only facilities in Malaysia we have only at the nuclear agency. So there's a lot of actually challenges because of the productions, because we need to send the sample at the nuclear agency, and they are not actually able to facilitate us. So in terms of that, there are challenges as well. But there are possibility for to scale up, because we have to rent the area in the nuclear agency, and we have to have a very big grants for that as well. And as mentioned, to scale up 2 million in Malaysia also not enough, because the facilities itself is very technicals, and that's why I'm exploring to the very less cost effective, like arabinoxylates. We can actually directly use the we can actually produce the polymeric membrane as such at our laboratories. Of course, in the challenges is the cost, and first and foremost, I would say.
Right, ma'am. Thank you so much. The second one is, ma'am, how can the natural polymers be optimized for controlled or targeted drug release in treating complex diseases such as cancer or neurological disorders?
Okay, if you say about the neurological, of course, the drugs might have to be to across the BBB, the blood-brain barrier. So, is it the you can actually protect the drugs until it can reach to the BBB or not? So actually, you can have to fine tune on the structure modifications in terms of cancer. Of course, you have to encapsulate the drugs, because of course you want to actually increase the cellular uptakes. So it's based on the polymer itself, and also you modify on the chemical structure that actually can prevent from the enzyme at that targeted area. Let's say at the area of the BBB, how you want to actually deliver the drugs into the BBB area.
Right, ma'am. Thank you so much, ma'am. The next one is, ma'am, how can natural polymers improve the solubility of poorly water-soluble drugs? Are there specific modifications that enhance this effect?
Poly?
Natural polymers can be improved for the poor water solubility.
Poor water solubility. Okay, maybe you can actually like poor water solubility of the drugs. Maybe you can actually encapsulate it with the polymer that actually not easily degraded. Okay, and then it can actually prevent from the degraded of the polymer into the gastro intestinal fluid, like actually in the stomach fluid especially. So when you actually can encapsulate it and then prevent from the degradation of the enzyme in the stomach fluid, so actually the drugs can enter the intestine. Okay, the drug can enter the intestines and it can be absorbed highly in the intestines. In terms of the structure of the polymer and also the drugs, you have to see what type of the drugs. Okay, what type of the drugs, whether the drugs is directly embedded in the polymer, or actually if it's low water solubility, it can be also is attached to the polymer. So you have to fine tune whether it's really that the polymer not interact with the drug itself and it can deliver to the target area.
Right, ma'am. Thank you so much, ma'am. The last question is, ma'am, how do the natural-based polymeric drug delivery system impact the immune system, and what steps are taken to ensure the biocompatibility?
Biocompatibility. You will say that the smaller size is better, because yeah, the smaller size is better because in terms of the macromolecule, of course it can induce a microphage. Okay, we can say the immune response. So when you actually reduce the size numbers of the particle itself, you actually can avoid from the immune response of the cells of the immune response toward the particles in the blood vessels. I would say that.
Right, ma'am. Thank you so much, ma'am, for answering all the questions wonderfully. And so here we come to the conclusion of this webinar. So as we conclude of today's 71st SPSR International Webinar on National-Based Polymeric Delivery System, enhancing therapeutic efficacy in pharmaceutical science. I would like to thank our esteemed speaker, Dr. Najwa Mohamad, all attendees, and the organizing team for their contributions to this enlightening session. Today's discussion have underscored the growing significance of natural-based polymers in creating advanced delivery systems that enhance drug stability, bioavailability, and targeted release. By leveraging the biocompatibility and biodegradability of these natural materials, researchers are paving the way for more effective and patient-friendly treatments that reduce side effects and align with sustainable practices. The potential of natural-based polymers in targeted and controlled drug delivery opens new pathways for treating complex diseases like cancer, chronic inflammation, and neurological disorders. As we look toward future applications, it's inspiring to consider how these systems can also support personalized medicines, allowing treatments to be tailored to individual patient needs. Today's insights reinforce the importance of continued research in natural-based polymeric systems to address some of the most pressing challenges in pharmaceutical science. This work not only advances therapeutic efficacy, but also aligns with the industry's shift towards greener and more ethical healthcare solutions. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all SPSR team members and participants for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We are grateful for your significant involvement in the 71st SPSR International Webinar. Your engaged participation and input have been essential, and we eagerly anticipate collaborating on upcoming initiatives aimed at enhancing healthcare and well-being for everyone. Once again, thank you, esteemed speaker, and all the attendees for making this event a success. We look forward to your continued engagement in future discussions. Stay connected, stay innovative, and together let's continue to enhance healthcare solutions outcomes for all. Have a wonderful day ahead. Thank you so much, ma'am.